Hello everyone, I'm Leilani Arisgado and today I will discuss to you about the prenatal development. A prenatal human is considered an embryo for the first 8 weeks, when rudiments of all body parts form. During the first week, the embryo is in a pre-implantation stage because it has not yet settled in the uterine lining. Some biologists consider a prenatal human to be an embryo when it begins to develop tissue layers at about 2 weeks. Prenatal development after the 8th week is the fetal period, when structures grow and specialize. From the start of the 9th week until birth, the prenatal human organism is a fetus. Sperm and oocyte meet at fertilization. Hundreds of millions of sperm cells are deposited in the vagina during sexual intercourse. A sperm cell can survive in a woman's body for up to 3 days, but the oocyte can only be fertilized in the 12 to 24 hours after ovulation. The woman's body helps sperm reach an oocyte, a process in the female called capacitation chemically activates sperm, and the oocyte secretes a chemical that attracts sperm. Contractions of the female's muscles and moving of sperm tails propel the sperm. Still only 200 or so sperm get near the oocyte. A sperm first contact a covering of follicle cells, called the corona radiata, that guards a secondary oocyte. The sperm acrosome then bursts, releasing enzymes that bore through a protective layer of a glycoprotein, the zona pellucida, beneath the corona radiata. Fertilization or conception begins when the outer membranes of the sperm and secondary oocyte mate and a protein on the sperm head contacts different types of protein on the oocyte. A wave of electricity spreads physical and chemical changes across the oocyte surface, which keep other sperm out. More than one sperm can enter an oocyte, but the resulting cell has too much genetic material development to follow. Fertilization by a sperm cell includes the oocyte into complete meiosis. Before fertilization occurs, the sperm acrosome bursts, spilling enzymes that help the sperm's nucleus enter the oocyte. Usually only the sperm heads enter the oocyte, wherein 12 hours of sperm penetration, the ovum nuclear membrane disassembles, and the 2 sets of chromosomes, called pronuclei, approach one another. Within each pronucleus, DNA replicates. Fertilization completes wherein the 2 genetic packages meet and merge, forming the genetic instruction for a new individual. The fertilized ovum is called a zygote. The biotics reading discusses cloning, which is a way to start development without a fertilized egg. The embryo claves and implants. A day after fertilization, the zygote divides by mitosis, beginning a period of frequent cell division called cleavage. The resulting early cells are called blastomeres. When the blastomeres form a solid ball of 16 or more cells. The embryo is called a morula, Latin for mulberry, which it resembles. During cleavage, organelles and molecules from the secondary oocyte, cytoplasm still control cellular activities. But some of the embryo genes begin to function. The ball of cells hollow out and its center fills with fluid, creating a blastocyst. The term cyst or cyst refers to the fluid-filled center. Some of the cells form a clamp on the inside lining called the inner cell. The inner cell mass. Its formation is the first event that distinguishes cells from each other by their relative position. The inner cell mass continues developing, forming the embryo. This picture is cleavage from ovulation to implantation. The zygote forms in the uterine tube when a sperm nucleus fuses with the nucleus of an oocyte. The embryo forms during the second week of prenatal development. Space called the amniotic cavity forms between the inner cell mass and the outer cells anchored to the uterine lining. Then the inner cell mass flattens into a two-layered embryonic disc. The layer nearest the amniotic cavity is the ectoderm. The inner layer, closer to the blastocyst cavity, is the endoderm. Shortly after a third layer, the mesoderm forms in the middle. This three-layered form sandwich-like structure is called the primordial embryo or the gastrula. The cells that form the layers of the primordial embryo, called primary germs layers, become determined or fated to develop as a specific cell types. The cells is specialized by the progressive switching of all the expression of genes important and active in the early embryo as other genes begin to be expressed. This picture is the human embryos and 28 days and 49 days. This is a 16 weeks fetus. A fetus at this stage has hair, eyebrows, lashes, and other features and look human.